Welcome to the video for chapter 12.1. We are in chapter 12 and we're going to talk all about viruses. Now, I'm going to give a caveat here. The field of virology is a whole science unto itself. You can take whole classes on just viruses, the different types, different shapes, what we know, how to prevent the spread. Everything can be covered in a virology class. It's actually one of my favorite classes that I have taken to date. Was my virology classes in both undergrad and in grad school. Now, we're going to kind of dip our toes a little bit into the virology pool of chapter 12, but we're going to mainly be talking about some specific examples of viruses and some overall virus characteristics. We're going to talk about how we classify them in different ways. So let's get started. So chapter 12.1 is all about how viruses infect host cells and all about their genomes and how we classify viruses based on their genomes. Okay, so our chapter 12 objectives are you need to be able to define the nature of a virus and you need to be able to explain how a virus differs from a cell. So our living versus non-living debate is coming back, so that's exciting. We need to be able to describe the major classes of viral genomes and be able to cite an example of each. We need to be able to explain how viruses are cultured. Not very easily. That's the answer. And then you're going to be able to describe the replication cycles of some very specific viruses. So we're going to talk about papillomaviruses, specifically the human papilloma virus that you guys know by HPV, the influenza virus, and the human immunodeficiency virus, or HIV. Okay, so as always, let's start the chapter with a case history. And our case study is a virus from the needle stick. So we are going to sunny Phoenix, Arizona, to a hospital there, where we have Christine, who is a newly graduated, newly minted 25-year-old critical care nurse. Okay, so Christine is going through a routine sample collection from a patient. One of the differences is this patient has a hepatitis C infection. So while Christine is trying to take the sample, something happens, and she accidentally sticks herself with the needle that she's using to collect the blood sample from her patient. This is not good. And unfortunately, this happens quite often when working with sharps like needles, especially in the healthcare field. It happens more often than we'd like to admit. Okay, so as soon as this happens, she reports it to her superiors, and she's put on a watch to look for any signs and symptoms that she also has caught this virus. Okay, so about 6 weeks after the initial stick, Christine develops a fever and a flu-like symptoms. Now, this could just be the flu, but because she had this accidental needle stick 6 weeks prior, the doctors are going to be a little bit suspicious if it's actually the flu. So one of the things that they're going to do is they're going to bring her in and test for hepatitis C. Okay, so they're starting to suspect that maybe this virus moved to her liver. Okay, so one of the things that Christine's doctors order is the first thing is she's going to do is give a blood sample. So we call these blood titers when we're looking at virus levels. Okay, so her blood titer comes back that she has the viral particles for the hepatitis C in her blood, and but they're fluctuating, they're going up and down. Okay, they're also going to test how well Christine's liver is functioning because hepatitis C is known to affect the liver. Okay, her liver function test also has levels that are going to fluctuate. Okay, so obviously Christine's immune system didn't kick in. She has caught the hepatitis C virus from this accidental needle stick. Okay, so what they're going to do is they're going to put her on a treatment of different antivirals. Remember, we can't use antibiotics with viruses. They only affect bacteria. Okay, so she was put on a course of ribavirin and an interferon alpha drug. Okay, so ribavirin is a RNA synthesis inhibitor, so it makes it so that RNA can't be made. And the other one is going to interfere with a specific protein that stabilizes what is called interferon in our bloodstream. Now, I wanted to, before we move to the next slide and talk about more of Christine's treatment. This is an example here on this slide of a healthy liver. So this is what your liver looks like if you're healthy. So those that have hepatitis C, their livers are going to look a little bit different because they're going to sustain some damage, which we call cirrhosis, um, in the course of their infection. So this is what a liver of an individual with hepatitis C looks like. So this is what we call cirrhosis of the liver. This is kind of where Christine's headed if they can't get the virus under control. Okay, so on the treatment, her viral levels are going to decrease to a very low level. Okay, in fact, we can only measure them with something called an RNA assay and using PCR to amplify it. Okay, the thing is, the treatment was working great. It was bringing the virus levels down. The thing is, the side effects of antivirals are pretty terrible. Okay, and because they were so terrible, Christine opted to stop the treatment instead of continuing it and getting rid of the virus altogether. So she discontinued therapy because the side effects of the therapy was actually worse than having the infection. Now, the thing is, even though her viral levels are low, Christine has the chance of her hepatitis C infection coming back. So she's good for about another decade. So about 10 years later, Christine has an unexplained fever. Now, because of her medical history, one of the first things they're going to test for is hepatitis C. Has her viral levels increased since 10 years ago? Okay, so she gives a blood sample, a PCR test is ran, and it tests positive for hepatitis C viral RNA. Okay, so they do put her on a similar treatment with the interferon and ribavirin, but just like last time, the side effects were too severe, they were too great, and they weren't as effective as the first treatment. Okay, which makes sense. 10 years is a long time. Viruses can adapt. Okay, they can evolve. So over the next 15 years, because she opted to go off the treatment again, Christine sees her liver function decline and decline and decline, and is showing severe cirrhosis. Okay, so in 2015, things are getting pretty bad. So Christine's at the point where she needs a new liver. Okay, but she might not make it until there's a donor that's able to give her a liver. So she does learn that there are 2 new antiviral drugs out. So I'm not even gonna try to say these guys. You can read the slide. Okay, and the point of these antiviral drugs is that they will stabilize her liver function and kind of hide her by until a donor can be found. But the problem with these drugs is they're new, meaning the company that developed them are still paying off the research and development to make the drugs. They're not tested as well, so there are side effects, which is also why they're expensive. Because of their expense, Christine doesn't know how she's going to pay for them, even though she needs them desperately. So this is a common problem in healthcare. Those who run research and development on new pharmaceuticals, they're very specialized in their field. They're very knowledgeable, and they want to be paid for that knowledge. And then the fact of running the experiments that are going to help to develop that drug also costs money. So pharmaceutical companies need to get back their investment in this research. Now, are there ways that we can make it cheaper? Probably. This is something I don't have an answer to. Most of the field doesn't have an answer to. But it is what it is at this current time, and hopefully soon we can figure out a way to solve this problem. So, because this is a chapter on viruses, let's talk about the hepatitis virus, specifically the hepatitis C virus. Now, there's multiple different viruses that can cause hepatitis. So hepatitis is actually a general term that just means that the liver is inflamed. Okay, so with hepatitis C, the inflammation is caused by damage from a virus. Okay, so we define a virus as a non-cellular, no cell membrane, no cell envelope, okay, with a genome contained by a capsid. So capsid is going to be like a little protein jacket, a little protein coat that's going to protect the genome from things that could degrade it. Okay, so we're going to get into the living versus non-living. So one of the reasons why viruses are mostly accepted as non-living organisms is the fact that they can't reproduce new viruses on their own. They have to use a host cell. Okay, so a virus is going to take over the host cell to use the cellular machinery of the host to make new viral particles. Okay, so some viruses like hepatitis C are going to have a RNA genome. Okay, so this is something that we don't see outside of living things. Most living things have a DNA genome. So we have other viruses like the herpes virus that do have DNA genomes. So with viruses, we can have either or. Now, with hepatitis C, we are an RNA virus. It has a very high mutation rate, which means that it's really hard to treat with antivirals. Okay, so a lot of times, like we saw in Christine's case, they're going to actually prescribe a drug cocktail because it's going to be harder for the virus to evolve to get to be resistant to the whole cocktail very soon. Okay, so if we administer one antiviral, the virus can mutate very quickly and develop resistance to that drug. Now, if we prescribe two antivirals, it's going to be harder for that virus to gain resistance to both at the same time. So one's going to work while the other doesn't. Okay, and if we prescribe a 3 antiviral drug cocktail, that is going to have even a less likely chance of the virus becoming resistant to it. Now, there are about 60 million chronic hepatitis C infections worldwide, and about 15% of the infections that are reported every year get cleared completely, but the majority of the population that get the hepatitis C virus are going to live with that virus for the rest of their lives. Now, the thing with antivirals, one, antivirals are expensive, and as we saw in Christine's case, they can have very severe side effects. Now, one of the reasons for this is a lot of antiviral drugs, the reason they work on viruses is they're going to take advantage of the same parts of the virus that allow it to hijack your cells. Okay, so because it takes advantage of these unique properties of the virus, you're essentially also going to attack the parts of your body where that virus can bind and hijack your cells to replicate. So this is why they have very severe side effects. So for example, the ribavirin that Christine was on, it inhibits red blood cell function. So it makes it so that you don't carry as much oxygen. So you're going to be anemic and you're going to be very tired and very just not functioning well. Okay, so that's a little bit more about hepatitis C. This is one that we watch out for a lot in healthcare. Now, so that's the case study. Let's talk about section 12.1. So we're going to talk about how viruses infect cells and cause disease. And we're going to talk about viral genomes. Now, we're going to talk about how we classify viruses based on their genomes, and we're going to look at some specific examples of each. But we're also going to talk about some vocabulary here. We're going to talk about what it means for transmission and something called a host range. And you're going to need to know these definitions, guys. Okay, so viruses, most of us in the scientific field classify them as non-living organisms because a virus is literally just a collection of genetic code surrounded by a protein jacket called the capsid. Now, they can be either DNA or RNA genomes, it doesn't matter. Doesn't matter if they're DNA or RNA, they're still non-living. Okay, they don't have any cell structure, they don't have any organization, they're not ordered. Okay, they don't have a cell membrane, they don't have they don't have ribosomes, they don't have anything to help them replicate themselves. Okay, they have no nucleus, nothing. Okay, so this is the main reason that other than them not having cells, the fact that they can't reproduce on their own is one of the main arguments for classifying them as non-living. Okay, if they are not in a living cell, the virus is inactive. They can't make more virus. They can't cause more infection. Okay, so they have to be inside a living host in order to do so. Okay, so viruses are going to hijack your cells. They're going to trick their way in, and they're going to use parts of your cell in order to replicate themselves to make what we call viral particles. Okay. Often this happens. A lot of viruses often kill the host cell in the process of replicating themselves, which will then cause damage to the host, as we saw with hepatitis C, because when this was happening, they were killing those liver cells off, which was causing the cirrhosis. Now, there are other viruses that are going to infect a host, but not kill the host, which is very advantageous for that virus because it then has a host for a longer period of time than the ones that actually kill their host. Okay, now, so let's talk about how viruses infect people and animals and things like that. Just like bacteria, viruses are ubiquitous. They are found everywhere. Okay, we have identified viruses that infect every taxa group, so every phyla, a group of organisms from bacteria to eukaryotes to archaea. So no living organism is unaffected by viruses. Okay, we have a lot of human diseases that are caused by viruses. So things like the common cold is a virus, the flu every year is caused by a virus. And then we have some specific types of viruses that can integrate into the genome and disrupt your genome and can cause cancer. We're actually going to talk about a specific case for this later in chapter 12. But the thing is, in nature, in natural environments, viruses have a very specific role to play. They're a very important part of our ecosystems. Now, what they do and how they play that part is different depending on the virus, but we do see this effect on nature when we remove viruses altogether. Now, does that mean that we can't get rid of viruses? No, we need to get rid of some of them. In fact, we've worked on getting rid of things like smallpox. Okay, we almost had things like measles eradicated, but we'll talk about that here in a second. So when we say viruses are ubiquitous, viruses are literally everywhere. We have found them everywhere. We have even found them in the polar ice caps up in the Arctic Circle, which, we found these ancient viruses in the ice and scientists reanimated them because science, apparently. Um, Researchers estimate that viruses are going to outnumber bacteria by 10 to 1, and we already know how much bacteria outnumber humans. So there's a lot of viruses out there. Now, because viruses don't have that cell wall, they don't have that peptidoglycan layer, we can't use antibiotics to treat them, which is why we have to develop these antiviral medications. But more often than not, instead of antivirals, we're going to actually develop a vaccine that's going to reduce the severity of the viral diseases. Okay, it doesn't prevent us from catching those diseases. It prevents the severe symptoms of those diseases. So we have vaccines that work for COVID-19. This is our latest one. We have vaccines for measles. We have vaccines for smallpox. We're working on HIV vaccines to prevent AIDS. So there's a lot of different vaccines in play, but we also have a lot of different medications. So we have antiviral medications that prevent people from catching HIV that will lead to AIDS. So we'll talk about a little bit about that as we go through this chapter. But this is why it's important for us to understand how viruses work and how to treat them, because they are literally everywhere. And you probably know somebody in the last 2 weeks who has gotten some form of virus and gone down sick. Okay, so let's talk a little bit about how viruses replicate. So we know that they can't copy their own genome, they can't make new viruses on their own. This is why they need a host cell. And because viruses are going to affect all sorts of living things, let's talk about a simple example that we can actually visualize what's happening here. So let's talk about bacteriophages. These are the viruses that infect bacteria. Okay, sometimes we refer to them as just phages and not bacteriophages because scientists are lazy and sometimes we like to shorten things. Now, with bacteria, it's a little bit different than, say, a human host cell and the viruses that get into human hosts. Bacteria don't have organelles. They don't have a nucleus. All of their transcription and translation happen in the bacterial cytoplasm. So as soon as the phage gets their genome into the cytoplasm, the bacteria can then replicate it and make proteins from it and start to make new progeny of that virus. Now, the progeny of a virus are referred to as virions. These are going to be completed viral particles. So when this is happening, one of the ways we start to study bacteriophages in the lab is we are going to get our plate and we're going to grow a lawn, just like we did in lab with lab 5. Now, when they add the bacteriophages, what we're going to see is we're going to see these dark spots, these are called plaques, develop in the lawn. Okay, so these dark spots indicate places where the bacteriophages are killing off the bacteria, which is why there's spots in the lawn clearing up. Okay, so that's one example of how viruses replicate in cells. So let's talk about another example. Let's talk about a virus that infects human cells. So let's talk about the measles virus. Now, the measles virus is interesting. We have a vaccine that works against the measles virus. And it worked so well that we almost had measles eradicated. So we only have one virus that we've completely eradicated, and that's the smallpox virus. Nobody gets smallpox anymore. Up until 10 to 15 years ago, there was an occasional measles case, but not very many. But one of the things that has happened in the last 10 or 15 years is the rate of vaccination. So how many people that are getting the measles vaccine has declined in the last 10 to 15 years due to a lot to these anti-vaccine movements and the flawed thinking that viruses cause things like autism, which they don't, been proven many times that they don't. Okay, but because of this decline in the vaccination rate in small children, we're also starting to see the rate of measles infections increase. Okay, in fact, one of the biggest measles outbreaks we've had in the last 10, 15 years happened because a group of homeschoolers that happened to be part of an anti-vaccine movement went to Disneyland. Okay, so children that were vaccinated against the virus, they wouldn't show any signs or symptoms because the vaccine was working. But these children who hadn't been vaccinated didn't have that protection, and so a lot of them got sick with the measles. Okay, the interesting thing about the measles virus is that it's what we call an enveloped virus. So it's going to take part of the host cell's plasma membrane with it to protect it from the immune system when it escapes the host cell and goes to infect another host cell. Okay, so we can see in this picture, okay, this is what measles look like. It looks like a rash. Okay, so here we can see the virus capsid. So we see this protein jacket, and then we see the envelope that it stole from the host cell right here. Okay, so we have a bunch of different viruses. One of the interesting ones that I've heard about in the last few years is that there's a virus that is affecting Tasmanian devils down in New Zealand and Australia, which is actually causing them to grow tumors on their face, and the placement of the tumor growth is interfering with the use of their mouths, so they can't eat. This is actually a really terrible problem because of this virus and the fact that the Tasmanian devils can't eat. It's actually starting to wipe their species out. So if we can't fix it anytime soon, we could see the extinction of Tasmanian devils happen. And then we have the elephant in the room, right? The SARS-CoV-2 virus, COVID-19, is the virus that a lot of us are very familiar with because of the pandemic. Okay, the thing is, all of these viruses share, whether they affect bacteria or humans, they all share the fact that they have to hijack the host cell in order to replicate. Okay, so one of the things we have to talk about is some vocabulary that goes along with talking about viruses. So we talk about transmission, host range, and tissue tropism a lot when we're referring to viruses. Now, transmission is the process of spreading viral particles, so helping viral particles get to a new host. So different viruses are going to be transmitted in different ways. We have HIV that's transmitted by blood or sexual reproduction fluids. We have viruses like the measles virus and the COVID-19 virus are transmitted by respiratory droplets in the air that get inhaled. And one of the reasons why they have very specific transmission processes is because they have what is called tissue tropism. So tissue tropism is the range of tissues that virus can infect. Okay, so where can that infection take place in your body? Now, a lot of viruses have very specific tissues that they can infect. So measles and COVID-19 are going to infect your respiratory system because they're transmitted by respiratory droplets. And one of the reasons why is because viruses have to be able to hijack a host cell. Okay, so they're going to only infect cells that they can gain entry to. Okay, they're going to use the proteins on the outside of their capsid, and they're going to be specific or close enough to specific molecules that bind to proteins in our receptor proteins on the outside of our cells as is. This is how they trick their way into the cell, is by binding to these receptor proteins. So they have to have specific receptors in these tissues in order to gain entry. So this is why we have tissue tropism, because they can only infect tissues that they can gain entry into those cells. Now, all viruses also have a host range. Okay, so how many species can that virus infect is what the host range is. Some viruses have a host range of 1. So HIV, human immunodeficiency virus, is a very narrow host range. It can only infect humans. It doesn't infect any other animal species or anything. Okay, but then we have viruses like the influenza virus that has a very broad or wide host range. It can infect lots of different species. So we have the bird flu, the swine flu, we have human flu. This is an example of a virus that has a very large host range. Okay, so we're going to be referring back to these terms as we go through the rest of chapter 12, and these will be on the next exam, guys. You have to know what these mean. Okay, and we'll talk about them as we start talking about very specific types of viruses as well. Okay, so let's talk about how viruses infect a cell. Okay, so viruses have very small genomes. Their genomes roughly are between 2,500 and about 2 million base pairs, depending on the size of the virus. So things like the panda virus is going to be on the larger side. So, and then other things like what we call the yellow dwarf tobacco virus, tobacco mosaic virus, is going to be around that 2,500 base pair range. Okay, because viruses have relatively small genomes compared to their host cells, that means that they can replicate more quickly. Okay, and they don't use a lot of energy to do so. Now, these viruses are also always going to contain genes for the proteins that are going to allow them to gain entry into the host cell. Okay, so if we look here, we have our virus particle. It has bound to the receptors on the outside of the cell. Okay, so it's attached to it. And because it's bound to these receptors, what it's going to do is it's going to trick the cell into thinking it's something it needs. It needs to bring this substance in. So this is how the virus is going to gain entry into your cell. Okay, so what they're going to do is they're going to break apart and they're going to release their genetic material. Okay, and when they do this, your machinery, so the things that do transcription and translation, are going to pick up this genetic material and it's going to start to replicate it and put it through transcription and translation, which means that you're going to make more of the viral genome. You're going to make the proteins that make up the virus, that capsid, that protein jacket, and you're going to make also proteins and enzymes that are going to help assemble these new virions, these new viral particles, and then going to help them get released from the cell. Now, some of them will just what we call burst out of the cell. They're going to lyse the cell. Others are going to take advantage and take part of that plasma membrane with them and create their own envelope. Okay, so because of this, and the fact that viruses have very small genomes and they only code for a very small number of proteins, this is why we don't have a lot of antivirals out there, because we can't really build antiviral medications because there's not a lot of things to target. This is why they're hard to discover, and it's also one of the reasons why they have severe side effects, because the only things that we really have to target with viruses are the same things that they use to manipulate their way into our cells. So we're going to affect our cells as well with these antivirals, which is why they have very severe side effects. But also because viral genomes are small, they replicate quickly and they have high mutation rates. Okay, so they're going to mutate really, really quickly. And so one antiviral that worked one week might not work a month later. Okay, they mutate more quickly than bacteria, which is why we go through several different versions of the flu every year. So you may get a flu shot and that might work for that strain, but because it mutates so quickly, usually you're going to catch the flu a few times even with the flu shot. Just hopefully the symptoms are less severe than if you had not had this flu shot. Okay, so the last thing we're going to talk about are viral genomes. So this is actually a really important piece of virology because we classify viruses based on their genome. So viruses can have genomes that are composed of DNA or RNA, depending on the virus. We can have double-stranded DNA. We can have single-stranded DNA. We can have single-stranded RNA, which is what we're more familiar with our mRNA. But we can also have double-stranded RNA, which makes sense, right? Because if we can have double-stranded DNA and it's complementary, it makes sense that we could have double-stranded RNA. Now, we don't see that as much as we see single-stranded or double-stranded, but it's still out there. There's still these viral genomes that have it. Okay, so the capsid, that protein jacket, is going to protect the viral genome and keep it intact. Okay, so one of the ways that we classify viruses is by what we call the Baltimore model. So the Baltimore model is named for David Baltimore. He's the guy that came up with this classification system. And the Baltimore model is solely based on the genome of the virus. Is it RNA or DNA? Is it single-stranded or is it double-stranded? Okay. Is it can the strand directly encode proteins, or does it require synthesis of something else that will help it encode the proteins? Okay. And one of the things that these proteins are going to do is that if a capsid is going to be encased by an envelope, okay, so that plasma membrane it sells from its host, what the virus can do is it can have proteins made using your cellular machinery that can then be embedded in that cellular membrane, and it's going to give this viruses different advantages. Okay, so it's like a spike protein. Could be a protein that's embedded in the envelope in this cell membrane from the host. Okay, so this is the Baltimore model. Okay, we have 7 groups. Okay, and each group has a different genome that it's going to classify. So we have group 1, which is our standard double-stranded DNA. So these are things like the herpes virus. Okay, they can directly translate the double-stranded DNA to mRNA and make proteins that way. We have group 2, which is going to have single-stranded DNA. These are going to be things like parvovirus. We see this a lot in dogs. Okay, so the single-stranded DNA is going to be replicated. The cellular machinery is going to make it double-stranded. Then it's going to go through and transcribe and translate it into proteins. Group 3 is going to use double-stranded RNA. It is a thing. Okay, so these are things like rotavirus. Okay, the thing is, to go from double-stranded RNA, you're going to need to have RNA-dependent RNA polymerase in order to do that. So you're going to take this double-stranded RNA and you're going to make it into mRNA. Okay, using that enzyme. Now, group 4 is single-stranded positive sense RNA. Okay, so this is things like COVID-19, the family of coronaviruses. They're also going to use RNA-dependent RNA polymerase and to directly make their mRNA. Now, we also have group 5, which are similar, but it's going to be single-stranded RNA that is what we call antisense or negative sense RNA. Okay, so this is the example of this is the flu virus, the influenza. We're also going to have this RNA-dependent polymerase that's going to make it to mRNA, but we're also going to need an enzyme that takes it from the negative sense and changes it to the positive sense. And then we're going to have it go back to the negative sense once we package it into the capsid when we're making the virions. Okay, then we have group 6. These are our retroviruses. These are the things like HIV. So they are negative sense single-stranded RNA. So what's going to happen is we're going to use an enzyme called reverse transcriptase. It's going to take this single-stranded RNA and convert it to double-stranded DNA that is then converted to mRNA and used to make the proteins needed to build the virions. Okay, in group 7, we're going to start with double-stranded DNA. So these viruses are going to make mRNA, and then once the mRNA is made, we're going to use reverse transcriptase to go back to double-stranded DNA for the viral genome. I don't really know a lot of examples of this one. This is a new one compared to when I went to through virology, but it's kind of cool. So one of the last things I want to talk about is what do I mean from the Baltimore model when I say positive sense or negative sense? So a lot of it has to do with the direction that the RNA is in when it's bundled into the viral genome. So positive sense or sense RNA is going to be from 5' to 3'. So this is very similar to mRNA. Because it's similar to mRNA and it goes from 5' to 3', remember replication always goes from 5' to 3', we can directly translate it in the host cell. Okay, whereas with negative sense, or what we call antisense RNA, this is going to be in the opposite direction. We're going to be 3' to 5'. Okay, it's going to be complementary to mRNA, so it can be converted to positive sense RNA by RNA polymerase, but that has to be done before it can be translated. It can't be directly used. Okay, so we have to go from negative sense to positive sense, and then it can be translated directly by the host cell. Okay, the thing is, we're going to use RNA-dependent RNA polymerase as the enzyme that does this process. Okay, so it's going to be the one that takes our negative sense and makes it positive sense. Okay, and makes it so that the host cell can use it. Okay, so that is 12.1. So in the next few chapters, next few sections, we're going to be talking about different viruses and different virus properties. So we'll see you for lecture 12.2.